Nathan, what else is coming in the pipeline? We obviously have the cytotoxic, the immunotherapy, we have the targeted therapy, a little bit of radiation. We don't use a lot of radiation in large cell lymphoma, though we should talk about this in a second. But what else you see coming that is?
Yeah, so the good news, I think it's probably good news, is that there's a lot of new targeted drugs hitting different types of targets that are being developed. A lot of these are bispecific antibodies. There's a couple of these bispecific T-cell engagers that are currently in late stage development. In fact, there's a plenary session at ASH looking at responses with bispecific in CAR T-cell failures. Small study, but the data looks pretty good for the CAR T-cell failures. There are
very durable CRs, yeah?
Yeah, and so I think for me, probably one of the most exciting of the targeted drugs are these kind of bispecific antibodies that potentially could do similar things that we see with CAR T cells, really, only they're using your own CAR T cells, drawing them to the tumor, and hopefully inducing this kind of T cell response. There are also other conjugated antibodies. There are some conjugated antibodies against other targets. I've seen some recent looks at things with against CD47, CD79B. So there are some other interesting conjugated
antibodies. Don't eat me antibody targets.
Yes.
I think this is a time where we actually see huge development in this area of multiple compounds coming. And the challenge obviously will be how do we study and how do we move them forward. Here is that the new regulatory pathways using real-world data could actually help us to develop those combinations faster than we typically would. Sometimes it's difficult to understand the contribution of agents in the combination, novel agents. But if you have some real-world data about the single agent activity, you can actually use it to supplement and have some idea if your combination really is helpful. Also, with all the molecular diversity of the patients, some of those novel agents are limited to certain population of patients with specific molecular marker, like for example MYD88. So comparing the outcome between MYD88 mutated patients treated with any therapy versus the targeted agents is very promising. We actually very interesting session on innovation clinical trials on Friday afternoon, where we have folks from FDA and also statisticians. So we're talking a lot about how do we develop those new studies and what's the threshold of evidence now which we need to move it. But I think we are coming to an understanding that we won't be able to do randomized studies for all those agents. And there's a huge unmet medical need. And what we need to do is to work in a novel way to be able to move those drugs very quickly to the clinic.
Absolutely. I mean, I think that the idea of doing, first of all, we lose a lot of drug by doing a randomized because of the issues we discussed earlier. But we cannot do conventional trial the same way. We lose the trees in the forest all the time, and we need to be able to do a small trial based on the rationale and molecular rationale that's measurable, looking hopefully at metabolic response and not necessarily just clinical, and then to try to be able to make early decision. This is a real challenge because patients do well, better with large cell lymphoma, but large cell lymphoma particularly, but this is still a lymphoma that is increasing, and the mortality by lymphoma has increased still in spite of all the high-dose therapy and transplant. And you look at the lymphoma incidence has increased 500% in folks that are 80 years old and over, who are still in very good shape in Florida and exercising, so we need to make sure that we have options.